Cool. So what did you want to talk about today?
Well, I ship a lot of queens and raise a lot of queens, and I think some of the latest stuff that you've been working on was mainly queen stress, was it not? Like temperature related and things of that nature.
Yeah, that's right. Actually, the most recent stuff I've been working on has been drone stress tolerance. There was a paper that just came out this morning, um, which had a press release associated with it. And I just finished doing an interview with CBC. So the
Oh my
latest,
gosh, did
latest
you?
stuff is actually drones. But yeah, the bulk of the work, and I think that the probably the more impactful and useful work has been on queens so far.
Yeah. What do you want to say anything about the drone findings? I deal with a lot of drones just because I do instrumental insemination and they are incredibly fragile. Some people, like I learned from Sue Kobe, and sometimes she would bank them like in a little drone frame that would hang down into the colony. But I got to where, maybe Friday night, because I keep a weekly insemination schedule, I got to where I'll just catch them when at their peak flight time, which varies according to the temperature on Friday. And then I'll just stay up late and knock it out because it seems like they don't improve with age like wine. What'd
Yeah.
you find out in that paper for your research?
Yeah, well, there were kind of two major findings. One is obvious to anybody who has experience with bees. It's like you say, they're just very fragile. There's surprisingly little really like hard data comparing drone stress response to worker stress tolerance, I should say. So part of it was just like establishing that it's like something that we're all familiar with, but it's just good to really have in the literature.
For sure.
And then the second part was though like looking at what's going on in their bodies because like given that drones are so sensitive and it's like disproportionately sensitive too, right? When you think about their body size compared to workers, you would expect all else being equal, an animal with a larger body should be more tolerant to temperature fluctuations and more tolerant to chemical exposure. Um, but they're not, they're less tolerant. And so our hypothesis was that that might be because, well, one way to explain that is if drones had lower levels of the kinds of proteins that help mitigate stress. So like if they had lower levels of detoxification enzymes, lower levels of heat shock proteins, that kind of thing, then that could explain why they might be more susceptible. But we found the exact opposite when we took
Really?
samples of their blood of drones and workers that had been or had not been stressed in different ways. And pretty pretty much across the board, there were a couple exceptions, but by and large, the drones actually had higher levels of stress response proteins than the workers, which was very confusing. Um, and
Gotta love
then,
science.
yeah, exactly. So the way we explain our way out of that one is that one of the proteins that shows the biggest difference between drones and workers is this protein hexamerin, which is kind of like, you can think of it as kind of like blubber, but for proteins. The fats are like a storage of energy in the form of lipids,
Hmm.
but and then so hexamerin is an amino acid storage protein, and those of
Oh,
course are
wow.
the building blocks of proteins. And drones have super low levels of this, like almost at the limit of detection, kind of low levels. And so we're sort of thinking like what it looks like is that they really have very little protein reserves. And so maybe it's like they've invested everything they have into having these proteins already present to be able to deal with a wide range of different stressors. So it's like hedging their bets, but then they have very little in reserve to like further deal with something really
Hmm.
acute when it comes along, is like the working hypothesis.
Yeah, I liked how you said explain our way out
Yeah.
of that. That's great.
Excuse me. Yeah, I mean, it just was kind of mind-boggling. It was like literally the exact opposite of what we expected to find. Um,
Yeah,
yeah.
and I was interested in how they mitigate viruses too, because I'm finishing up some classes, and they were looking at what was it? I don't know if it was KATP pumps and whatnot, and they were saying how that whole process basically can affect whether they have a higher viral resistance or not, or whether they can clear viruses, which I still haven't pieced that together in my mind. I know there's certain behaviors that help bees at a colony level get rid of viruses, like it seemed to me whenever I started using high VSH stock, there was something weird going on there where if I saw a deformed wing virus and I would replace it with a VSH queen, that the colony would clear the virus out. But then, well, this, like I said, gotta love science, like the Evans came out with a paper that said, they can pass it super easily back and forth. And it was kind of, I'm like, oh my gosh, something's off there. But then Kara Wagner said, no, that is correct. But there's some kind of a mechanism behaviorally where they clear out I guess they fall on the sword, so to speak, the ones that are removing the infected pupa. If they get infected too, there's something going on where they clear out because the whole viral levels of the colony plummet. But I thought it was cool too, because I was listening to an interview that you did. I think it was on maybe a scholarship award or something you had gotten. And you touched on, which it was queens, and it was heat shock proteins with the queens and how it related to being able to clear viruses, was it not? Did I understand that correctly?
Yeah, that's right. Some of our findings show a pretty strong correlation between the queens expressing heat shock proteins and their level of viral infection. So queens that are more heavily infected with viruses tend to express more heat shock proteins. And so then the question is like, are the heat shock proteins like helping to fight off the virus, or are they some kind of other downstream effect of pathogenesis? And we have done some infection studies now as well, and other people have done this too in workers, where you infect the bees with virus and you see elevated levels of heat shock proteins.
Really?
And then so my next question is, does it go in the other direction? Like if you heat shock the bees first, are they then resistant to the viruses? So like by sort of artificially priming them to already have high expression of heat shock proteins, can you make them be able to tolerate a subsequent infection? So Alex McMenamin and Michelle Flanagan did some work in workers similar to this, but they did it the other way around. They were working on workers, and they didn't heat shock the workers first. They infected them with the virus first and then heat shocked them and then looked at their ability to clear the virus. But what I really wanted to know was like, could you actually prevent the infection? So
with kind of a pre-treatment, pre-treatment kind of.
Yeah, exactly. Like you can, if you take the term vaccine to mean something generally that you can apply to prevent an infection, then you could think of it as like a heat vaccine.
That's cool.
Yeah, and so I don't have a definitive answer on that yet, because we had all kinds of problems rearing queens last year. Unfortunately, not only me, but also the commercial producer who I was then defaulting to getting queen cells from. We were all having problems. So I didn't have nearly as big of a sample as I wanted, but the trend is like just a hair not significant. Like it's very, very close to being to the heat treated queens having lower virus infection when they are then subsequently infected in the lab compared to the non-heat treated queens, but it's just not quite significant. So
interesting.
The trend is in the right direction though. So I'm going to repeat it this year, hopefully with a bigger sample and get a clearer picture.
That's pretty cool. And I was interested too when you were talking about sperm viability after they'd been exposed to temperature extremes. And obviously that's something anybody who males queens, whether they're aware of it or not, have probably been affected by that. And it got to where, which, you know, I do transportation logistics for my day job. So that's something I have experience with. I got to where I would just ship next day air because it seemed like for some reason ground, obviously the transit time's longer, but it was way harder on them, and I ended up replacing more bees that way. And then whenever I switched like the next day air, it was night and day different. And I'm assuming it has to do with, you know, temperature extremes or stress, you know, all the above, being out of the colony and out of their natural environment. But what was some things you kind of found on that? Didn't you have like I like the term, the Goldilocks zone, you know, where it's not too hot and not too cold?
Yeah, exactly. Yeah, so we this was work that actually Jeff Pettis was the one who spearheaded quite a while ago, and I just analyzed and interpreted the data.
Awesome.
So what he did was he took somewhere around 200 queens and exposed them to different temperatures for different lengths of time, all the way from I can't remember if it was 4 degrees or 5 degrees was the lower limit now.
Celsius.
Yeah, so like fridge temperature, all the way from that up to 42 degrees Celsius. And then what you get from that data is like a curve where there's sort of the maximal sperm viability somewhere around hive temperature, like you'd expect. And then at either end, it drops off. And so then the question becomes, like, where along that drop off then do you say is too much? And to answer that, then we looked at my queen survey data from many different beekeepers in BC contributed to this queen survey, and they gave me failed queens and healthy queens, and then I could see how much of a difference of sperm viability there was between the two. And we found that the failed queens, as you'd expect, had significantly lower sperm viability, and it was lower by about 11.5%. So then that was like the threshold that we used as the drop off, like how much of a drop is too much. We just drew a line at 11.5% and looked at which high and low temperatures that intersected with on the curve. The argument being that 11.5% is like, there's now, because of the survey, there was evidence that that's like an economically and biologically meaningful drop.
Sure.
Yeah, so when you do that, then that's how we end up with the 15 to 38 degree, like Goldilocks zone, as
I like that term.
Termed it. Yeah.
You may not know exactly, but part of that I saw, which blew my freaking mind. It was like whenever they were looking at sperm viability, and this may be a Jeff thing, but how did he fluorescently mark them to where the alive ones are green and the dead ones are red? How in the devil does that work?
Yeah, it's actually, yeah, it's something we do it all the time. And it's not nearly as complicated as it might seem. It's not
black
like if
magic.
Yeah, no. And, you know, you might've seen the images where it just kind of looks like a galaxy of red and green sperm cells. And you might look at that and be like, how the heck would they stain each one of those dead sperm cells red and like know that they got it right? But it's much simpler than that. All you do is you take a sample of sperm solution and you spike in 2 different fluorescent dyes, Propidium iodide, which is the red one, and Cyber, which is the green one. And the mechanism is that the live cells can pump out the Propidium iodide. So then they won't show up as red, but the dead cells can't do that because
it stains them basically.
Yeah, so it just stains them. That's right. Wow, so that's how you can get the differential staining.
That was really cool. Yeah, I couldn't conceptualize how something like that would work, but it's genius.
Yeah, it's very straightforward. You literally just like take a little bit of each dye, stick it in the tube, mix it for a few minutes, and then look at it under the microscope and it's ready to go.
That's really cool. Man.
Yeah, and on the note of shipping data, so we have published some of that. And what we've typically seen, and what other people have seen also, is that usually for shipping, it's the colder temperatures that you have to worry about more.
Really?
Yeah. So, for example, in the data we published, we saw anything from 4 degrees Celsius up to 38 degrees Celsius was the maximum in our data. And that was, I think that was only 7 or 8 different shipments that we looked at. But the 4-degree one was like the really concerning one. And then some other people, I believe this was Pierre's group in Quebec, has shipping data as well, where I think they didn't see such extremes in either direction there. And then Jeff's old shipping data saw a spike as low as 8 degrees. So both ours and Jeff's show that there are some low spikes happening. And there's a lot of unpublished shipping data too, which hasn't been formally analyzed yet. But in that, it also looks like it's around 10% of shipments that have some kind of a temperature event. So
was it mortality, or was it like loss of viability whenever it got really cold like that? Do you know offhand?
Well, Jeff's paper from 2016 showed that the most clearly. So they had temperature loggers in different shipments, and they also even tried taking like the same batch of queens and then shipping half of them by USPS and half of them by UPS. And
That would be cool.
They were lucky enough that for one of those shipments, they one batch, so I can't remember if now which service it was that had the temperature event, but one batch had the temperature event and the other one didn't. So then, because the queens came from the same source, they could check the difference in viability. And they did see that the one that experienced the 8-degree low spike did have lower viability. So it's very, very rare to see queen mortality. I'm sure it has happened. I haven't had that happen personally, but I'm sure it's happened out there. But it's much more common to have these kinds of stress events or temperature spikes, and the queens look like they're fine, but really some fraction of their sperm has died. So
they've been compromised.
I really liked what you said in the beginning about whether you know it or not, most beekeepers have been affected by this, because I think that this is having a bigger effect than we've been able to measure, just because, like, if a queen, say, has 5 million sperm stored in her spermatheca, and then she gets too cold for too long and, say, loses 20% viability, then she still has, if assuming all those sperm were viable in the beginning, she would still have 4 million sperm, which she'll look like a fine queen when you introduce her and evaluate her for the first while, but she will fail earlier than a queen that didn't have that loss of viability. And that's the comparison that we are almost always missing, is like, okay, she seems fine now, but how good could she have been? Right. And so the story is sort of that I hear a lot from beekeepers, and you can maybe tell me what the sort of sense is in your circle, but it sounds like queen failure is a pretty big problem for a lot of people.
Absolutely.
Right.
Yeah, it's gotten a lot worse, well, obviously since Varroa showed up, but probably associated viruses and pathogens, but overall it seems like everyone agrees that they don't last like they used to, even in a typical setting where they're not being shipped across the United States. That's just added stress on top of that. But yeah, it has a big effect. But with me, where I see, and I've seen mortality down here, it's bad whenever it starts getting hot. So it's the opposite. I'm not usually shipping queens whenever I say that. I had snow on my cell builders last year, but it went away quick. It's Missouri. You never know what's going to happen. But where I had mortality or replacements was closer to July. whenever it would get up close to 90 degrees or so. And then, you know, that's another thing. You were saying USPS versus UPS or the brown. You know, one thing that you obviously see is the color of their vehicles. You know, like USPS is white, so obviously they're going to stay cooler, even though they're slower, and you don't know where your crap is, you know, because their tracking's not nearly as good. And then brown, they're quick, they're efficient, tracking's really good, but their vehicles are brown, so you know the internal temperature is going to be higher. You know,
Yeah.
and oddly enough, the worst that I had was in the state of Missouri. Like I can ship all over the US. The worst problems I have are intra-state, which didn't make sense to me. But there's so many variables, you know, and transportation.
Yeah, when you say worst problems, you mean you actually had your queens die in transit?
Yeah, they would show up. Yep. KIA.
Wow.
Yeah. And, you know, I just do this for a side gig, and, you know, customer service is important. So I guarantee live delivery. If they show up dead, I just send new ones, no questions asked. You know, they'll send pictures or whatever, but it's consistently certain areas, you know, so it has to be the way they're handled or something odd's going on, which, like I said, there's a lot of different variables, but it would be cool to do temperature logger studies and things like that. I could get into that. That would be interesting.
Yeah,
But I just
it's
switched to next day air because it's quick.
yeah.
It's not as stressful on the queens. I drop them off before 5 p.m. and most of the time before noon the next day they're at destination.
Yeah.
But
Yeah.
yeah,
That
way
makes
less
sense. That's the way
stress.
to go. But it's
Mhm.
really a shame that the temperature loggers are so expensive. Because
Where can you get those? Like if you were going to
um
do some test shipments,
yeah,
I'm sure Google.
I ordered them online. I think that the company is called Watchdog. Yeah, that's what it is.
Okay.
Watchdog. Watchdog temperature
Does it just
loggers.
take a measurement every 30 minutes or hourly or something like that?
You program it, so it's they're a little bit finicky to set up, but there's a very basic like software package that you have to download with the temperature loggers, or to use the temperature loggers, and you program it to record either anywhere from, I think it's 10 minutes up to an hour intervals. So if you use, I believe if you use a 15-minute intervals, you get around 2 weeks worth of memory for that many data points in the logger.
Sweet.
So more than long enough to use for the purpose of shipment. But each one of those temperature loggers costs about $100.
Yeah.
Which seems like a lot for, it's just like the size of like a watch battery. What I would really like, and I've had this thought and have forgotten to look into it, but if there's such a thing as like a color indicator for temperature extremes, that would be super useful, because the problem with temperature loggers also is that then your recipient has to like promise to send it back to you.
Yeah. Right. Yeah.
Or you lose your hardware and it's like $100 out the window. And then you have to be the one to like download the data and look at it. But if there was such a thing as just like a pH strip, but for temperature, you know,
Wouldn't be surprised. I know they have some that, which they called it shock watch. So they would put it on
yeah, exactly,
TVs
something
and
like
stuff,
that.
and if something hit it, you know, they would know.
Yeah, that would be way better. Like it would make more sense to use something disposable for something like this, because I mean, you don't always know who you're shipping to. Even
Yeah,
for me, when
exactly.
I try to have a good line of communication with people that I'm shipping to, I don't always get them back. So
Yeah.
yeah, that's something to look into. I wouldn't recommend going the actual like temperature logger route, just because getting them back is such pain.
Indeed.
Yeah.
Man, I was reading. Did you have anything that you wanted to add on that, like with the queens and the heat shock proteins and stuff? Anything that high points that come to mind? I was about to squirrel on Asian hornets because I saw that article you'd written in January.
Yeah, I think we can move on to Asian hornets,
Okay.
although I'm like really not an expert in them. I've talked to other people who are experts, but I'm curious what you have to say about them.
Oh, well, I just random people ask me about murder hornets, because I don't know what they're watching on TV or what they're seeing, but it seems like there was a lot of awareness brought to it, just to the general population. I mean, we've had to talk about it like at our state B meetings and stuff, and I don't know, I kind of thought when they originally found the one nest, I thought maybe eradication was possible, which eradication is difficult. It seems like there's always an escapee somewhere, but I guess they keep finding more. And
Mhm.
I don't know. Do you know anything about how the climate or anything? I wonder if they would be able to eventually establish, because I know we have Europeans down here that look remarkably similar to those.
Yeah, I think based on where their native range is and what the climates are like there, I think that it is feasible for them to establish here. I think that's pretty widely agreed upon. And there was even the one study, I can't remember which paper it was in now, but it just based on climate data and where there were known nests located so far, they sort of modeled different distribution patterns that may happen, like where it may spread to and how approximately how quickly, although
Where
that's
it's
very
most likely.
difficult. Yeah, that's very
Sure.
difficult to say for sure, but just like as a what you might expect. So I think it's definitely feasible for them to establish, and I think right now the best estimates, even though only a few nests were identified, I think just based on where those nests were and where individual hornets have also been sighted, they're the few that have been identified are almost certainly not all of them. So then there's the other question of like, okay, even if there are still some out there that haven't been identified and eradicated yet, are there sufficient genetics in the population for it to be viable and sustaining? And that is a really big question. That's where probably you could hope that they might not be. It's not totally clear that they're here to stay yet, but I think that the risk is like sufficiently high that
It's pretty high. Yeah.
that
Man,
it's a
I
good
thought
idea.
I read
Sorry,
try to
that it's a good
eradicate.
idea. Yeah, to do as
Yeah.
much as you can to eradicate it.
I think I'd seen somewhere where it said they mate with just one male, which I thought was odd. I don't know how yellow jackets or, other vespids, I don't really know their, sexual habits, so to speak, but it seemed like it was a bit odd, with somebody that thinks of it in honeybee concepts, where there's 16, 20, estimates.
74. I think 74 is the highest number I've seen for honeybees.
Wow, that's
Yeah.
insane.
Yeah. James Withrow from the Tarpey lab, he had a paper looking at all different patrilines in colonies, and there was one that had, I think it was either 74 or 76. It was 70,
That's
70
wild.
something different drone
So
fathers
there's a
within
lot of
one
variation.
colony. So
Yeah.
yeah, there are a lot of different mating systems out there.
Right. Yeah. And they're basically in your backyard, aren't they? Did I lose you, Allison?
Oh,
Oh, there you're back.
yeah. I don't know
I
what just
lost you for
happened.
a second.
Yeah,
Technology.
it says my internet connection is unstable.
Uh-oh. We got lucky. I live in the middle of nowhere and they buried fiber optic up through here. So we have 1 gig per second fiber out in the middle of nowhere. It was like our
Wow.
power cooperative got a grant or something. So, and it was right before they moved me to work remotely. So the timing was good.
Yeah, that's convenient. I don't know why. It's a constant struggle here. There's I think there are only two like convenient internet service providers where I am here in Delta, which is just outside of Vancouver, and our reliability is not great.
Sketchy. I've seen a lot of people getting Starlink. I think that's something Elon Musk had come up with. like satellite internet. I've seen
Yeah,
a couple of people on my Facebook have within the last week or two hooked it up.
yeah.
So that may be an option for you if it gets too
Yeah,
crazy.
it just seems a little bit silly because like my dad has ExploreNet, which is satellite internet, but he lives on an island that is 9 hours away from here that is boat
Oh, is it
access
really?
only and
Is that
off
where
the
you were
grid.
raised?
And yeah, that's right. But like
That's
he has
awesome.
satellite internet and that makes sense. It doesn't make sense to live just outside Vancouver and have to
Right,
have
and have
satellite internet.
satellite. Yeah.
Yeah.
Well, one guy that got it, he was close to St. Louis. I don't know exactly where he's at. I mentally peg him as St. Louis, but yeah, man.
Yeah. But going back to the queen stuff, I think that a really important area of future research is going to be looking more into virus infections in queens. Um,
Completely agree.
it just like that's just where all the data is kind of pointing us to right now. And I think it's probably been a pretty underappreciated issue. I don't know if that's just because it's difficult to study or because queens are precious, but almost all of the virus data so far is really in workers. And I think, and we sort of assume that the queens are like protected from infections because they have that social buffer of like the young workers mainly tending to them, the ones that tend to be less infected themselves. So then they're like distanced from the riskier foragers. But yeah, it's our survey data is telling us that it's something worth looking into though, because
Oh, absolutely.
the failed queens were more infected. The failed queens had smaller ovaries. And then we did another experiment where we infected queens with a virus and compared their ovary size and found that the infected ones had smaller ovaries too. So then you
Really?
actually have a causal relationship there.
What was the
And
virus? Was it something in particular?
yeah, that one was IAPV.
Okay,
So
Israeli acute paralysis virus. Okay,
yeah, but in the queen survey data, we only identified 3 different viruses in the queens with any level of consistency. So those were deformed wing, sackbrood, and blackwood cell virus. And it was in that data that we originally saw this relationship between higher viral infection and smaller ovaries. And then to sort of see that reflected in an experiment using a different virus makes us think that it might be kind of like a bigger picture. Like it's not like one virus has this one particular effect on the ovaries and it makes them smaller. It's like just being infected, having a high viral load in general is like costing resources that
that makes
would otherwise
sense.
go into ovarial activation is like the running idea.
Yeah, that makes sense. I was kind of running through it in my mind too. And, but I mean, you think of if somebody's sick, even a chicken, if they're sick, they're not going to lay as many eggs. That's what I was wondering if ovarial size has to do with how active they are with oviposition, maybe. So if they're sicker and their body's not healthy, they're not getting the right nutrition, or your body's trying to fight infection rather than make eggs. Do you think that's why the ovaries shrunk, maybe?
Yeah, it's like the idea of there being a trade-off in
Mm,
resource use. So like going back to the drones, it's kind of a similar idea. It's like you can't, resource availability is limited. Like there's a finite amount of amino acids that you could invest in
mhm.
synthesizing new proteins, and you sort of have to like choose if you're going to have a broad, if you're going to produce a large number of proteins or like have some in reserve to specialize and produce others. But either way, there's a limited, a finite supply. And it's kind of the same idea, though in a totally different context with the queens, that there's a finite supply of resources to be able to invest in either egg production or fighting off pathogens. So yeah, that's called the reproductive immunity trade-off hypothesis.
Oh,
And
interesting.
yeah, so we're trying to get into the weeds of it now. We have like the really
Yeah.
high level survey data showing these kind of messy but still significant associations from field data, and now we're trying to get more into really the mechanisms of what's going on with doing these terrible experiments to queens in the lab.
It's for the greater good. I mean, otherwise, how do you know?
Yeah, you know, there are those funny the challenges of like explain your profession badly in one sentence or whatever. And yeah, mine
Yeah.
would be I torture queens for a living.
Mad scientist stuff.
Yeah, but
And I'm really interested in the viral side of things too. You know, like I mentioned before, I think there's behavioral mechanisms that, which I think that paper that you were on with Kirsten, it's been several years ago, but I thought that was a killer paper. But it kind of danced around that too, because I think it said that they had been documented like VSH strains of ejecting, what was it, VDV, maybe Varroa destructor virus, or maybe it was IAPV, something like that. But I think it said, but not deformed wing virus, which was probably based on someone's study. But I've seen ridiculous reductions in deformed wing virus too. So I would really like to tear into that more too, but I don't know. I don't know if I will or not. I sent some just viral load in general. Like I had sent some, because I had some test apiaries that were not treated, that were just for VSH selection. And Randy Oliver years ago wanted samples sent from untreated apiaries. I figured it was so they could go, oh my God, look at these insane viral loads. But it was weird because it came back, because they tested for A, B, and C strain deformed wing virus. And they said, it said in the description, note, something listed as BL was below quantifiable levels. And it said like A, below quantifiable levels, B, below quantifiable levels, and C, none, like it was non-existent. And those were in colonies that had not been treated, period. But like I said before, if I saw a colony that was ate up with mites, because I like to experiment, I would just replace the queen with a VS, like from an instrumentally inseminated VSH breeder queen. And within 6 weeks or so, completely cleared up. Like, you know, most people will requeen and put a caricide or miticide in it, but I just wanted to see what would happen. And like, I've seen it repeatedly, and not even in drones. So there's some kind of a mechanism there. Maybe like, you know, Kara Wagner was talking about unhealthy brood odor. You know, maybe that's how those bees are detecting sick pupae. There's something going on there because they're ejecting them. Because I couldn't figure out, and I didn't know the viral resistance mechanisms of the physiology of the bee, which I keep learning more about.
Yeah,
but I knew there was like something mechanical about it. It was like consistent.
Yeah.
But it would be cool if I could send samples and run studies and figure out exactly what's going on, and just dig a little deeper, have controls too.
Yeah, well, I mean, it does make complete sense for VSH stock to have lower viral loads in general, because you know, we always say we don't have any tools to fight viruses, but the best tool to fight viruses is to fight Varroa, right? So if you're reducing the Varroa infestation by that much, then maybe it's like it gives the bees enough of a chance to clear the virus on their own without getting like these reinfections and persistent spread in the colony. So that makes sense to me. You'd expect those two things to go hand in hand.
For sure. And is there, where would you, like where Randy sent them, I forget what the name of the university was. It was in the UK, but I wondered if there was anywhere in the United States that runs viral analysis on honeybees that, you know, is pretty reliable. and not
Yeah, I
in
mean, David Tarpey's lab does it.
Tarpey's lab?
through
Okay.
yeah through the NCSU apiculture program. They
What did you say it was? NCSU apiculture?
yeah, that's right.
Okay.
Yeah, they have what's called the Queen and Disease Clinic. So beekeepers can send queens and now drones as well to the clinic, and then they take all these morphometric measurements and things like sperm viability and sperm counts for both drones and queens. And then they can kind of compile all that data into a report card and tell you if your
That's
queen
really
is
cool.
like an A, B, or a C queen. So yeah, that's a really awesome service, and they also do pathogen screening as well. And I believe it's fee for service, and I have no idea what those fees are, but I imagine that it's on their website.
Yeah, I'll investigate that. That's pretty cool. I might be able to tie something together. But
Yeah.
I'm finishing up the master's. I'm supposed to walk May 13th, and like,
Oh, congratulations.
oh, thank you. I feel good about it. I mean, I really enjoyed it because it's entomology. I'm kind of an ADD kid, so if it's something I'm not interested in, it's really difficult for me to pay attention. But if it's something I'm interested in, I'm kind of obsessive over it.
I think that's probably true for most people.
Yeah, I think you're right. I think you're right. Yeah. I know that, even whenever I was in kindergarten, my mom, would talk to my teacher and she's like, well, he's smart. He likes to look out the window a lot, watch the birds.
Yeah.
But I don't know. You mentioned something too. I don't even know that I'll do this exactly, but you said something about, was it remote doctoral work maybe before that was a thing? Do you think that's
Yeah.
evolving? Because I've pondered potentially furthering my studies since my employer pays a fair portion of it, but it would have to be, like I said, something I'm interested in. So something to do with VSH, something to do with breeding, something that maybe involved instrumental insemination or viral analysis and picking apart some stuff we're not really familiar with. But I have no idea where to go or who I'd want to contact to see if they would include me and things of that nature.
Yeah. I think that, well, given that a lot of doctoral students for the last few years have been like specifically asked to work from home, it's definitely become a more normal thing to do. I think that where it would be a disadvantage is like if you're a new student or working with a new group who you don't already have strong connections with, then I think that you'd be at a disadvantage to
Agreed.
do like everything from home. But where I think it works well is like if you take the time to invest in like actually being at the new place and learning the new techniques and doing the work in the lab, and then like taking your data home to analyze. Or if you are really into informatics and are handy with computer coding and stuff like that, then
That's not me.
okay, that's too bad, because those projects are probably the ones that lend themselves the best to remote work. Yeah.
And what do you mean by that exactly, like computer coding?
Well, like there are just so many data sets that are publicly available now. Say like transcriptomic data sets where they have like different, I don't know, they've looked at different exposure conditions and then checked which genes are elevated or decreased according to the treatment groups and that kind of thing. There are just so many data sets like that, that there are more and more opportunities to like combine them in different ways to ask new questions. And for that kind of work, the data is all downloadable and exists already. So like all the bench work has been done. The stuff that would have to be done in the lab is already over. And then you just it would rely almost entirely on your ability to like come up with new questions, to new ways to analyze the data that that provide new insights that there wasn't the opportunity for individually, that kind of thing.
Yeah.
So
makes sense.
Yeah.
I'll roll it around. I'm not ready to jump into anything because it seems like I've gotten too busy. But my wife says I hate relaxing, so I'll probably be looking for something else to do. But the bees keep me certainly busy. But it's in the back of my mind because some people assume, they're like, "Oh, you're finishing your master's. Well, you're going to do a doctorate." I'm like, well, why would you assume that? But it's kind of in the back of my mind. I don't know. We'll see where it goes.
Hmm. Yeah, I mean,
It would
this
have to be a
part,
good fit.
yeah, yeah, exactly. And this part probably shouldn't be like posted online, but this little bit is just between you and me. But
Sure.
if you haven't already, the department at Washington State University might be a good place for you to reach out to. They they just, I don't know why. I just see you like fitting in with the work that they do there very well.
Yeah.
Like Brandon Hopkins, um, he did a lot of the work on like cryopreservation of sperm.
I've
And
gone down that rabbit hole recently too, because I
right.
learned from Sue and they were working on it some. And then I did a recording with Shabu Raj actually yesterday, and he is already a step ahead of me there and getting into cryo. And so I may actually be out there at WSU at the cryo conference that they've got coming up.
Oh,
I haven't
perfect.
committed yet, but
Yeah.
sorry, I think I cut you off. What were you saying about
No,
Brandon?
that's okay. My like final thought for that is just that they do a lot of extension work there as well, and they would probably feel lucky to be able to, at the very least, collaborate with somebody like you who has both a science background and beekeeping
Sure.
experience. Um, like more than just hobby beekeeping experience. So
Yeah,
somebody
I've kind
like
of
you
gone
would
off the
be,
deep end.
yeah, you'd be kind of like the ideal either collaborator or maybe they would consider something like a further remote degree.
That would be awesome. I may contact them and just see what happens, can't hurt to ask. It may not work out. If the door closes, it's closed, just
Yeah,
kind of the way it goes. But
yeah,
I've been hung up on VSH so long, but we started using like SMR or VSH assays in the past couple years. And I've gotten to where 100% of the breeders allow 0% mite reproduction, which I cheat because I use instrumental insemination, but it's just a matter of doing the assays and finding out who's got it and who doesn't, and then
hmm,
controlling mating. It's pretty simple as that. But the thing is too, is they're commercially useful. They're not worthless, tiny little single box bees that just live but don't do anything. I mean,
yeah.
they rock and roll too, so they're commercially viable.
Huh.
I think
Even
I
though
think
they're instrumentally inseminated, because that's usually a big criticism for
yeah,
instrumental
well,
insemination, right?
yeah, I pull honey off of a lot of our instrumentally inseminated breeders, but even the F1s, which they were open mated here, but most of those tested 0% mite reproduction too, even after open mating,
Right.
which is
How
kind of cool.
many
But they
how
were,
many generations do you find that your VSH trait lasts for? Like for your F2,
well,
would they still maintain super low
that's a
mite
good
levels?
question. I need to do more work on that because I don't ever graft out of anything that's F2. Occasionally I'll graft out of an F1, but usually what I'm doing is using breeders for the queens, and then I'll test a ton of F1 colonies, and then I strictly just catch drones out of those because the open mating's kind of irrelevant there. If their parents were fours, the drones are going to be fours, because they don't have a father, only a grandfather.
Yeah,
And
yeah.
so,
If you're the only beekeeper around.
yeah, there's a couple around me, but I'm pretty remote, and a lot of them will use my queens, not all of them, but there's no one really close to me. It's kind
Right.
of a crappy honey production area where I'm at, so it's not highly desirable, and
Well,
there's
that's
not
better
much
for
out here.
breeding though. So
Oh yeah, absolutely. And we've got two pretty reliable flows. Like I've got a fall flow that I can really get a lot of good queens off of too, which I don't do as much. And whenever you said your experiment was failing because the queen rearing was totally a bust, I experienced that a couple years ago, and
did you
it
find out
didn't matter
why?
what I did. it had to have been something environmental, because I even switched yards about 7 miles away and switched cell builders and everything, and I can always reliably produce queens, and they just all gave me the middle finger. Like
Yeah,
not
interesting.
even mediocre production. They refused. That just, I have no idea what happened.
Like
I've
you
never seen
were,
anything like it. It had to have been pollen or nectar shut off, to where there's no stimulation. But even if I put them like in a big colony in a queenless environment, they refused. It was, I don't
so
know. It was
sorry,
really
they were
odd.
refusing to draw your queen
Produce.
cells, or
Yep.
you had queen cells that then were not viable.
No, they wouldn't even draw queen cells, period. It was like
Hmm.
0, 0, 0, 0, 0. And I was like, okay, we're done here. Because
Yeah.
I'd even switched yards and switched cell builders. It was odd. It had to have been something environmental. I mean, they weren't sick or they hadn't been sprayed or anything like that, And like I said, it was separate yards too.
Yeah,
But
yeah, that is weird. When I saw it happening here, the first thing that I thought that it was probably black queen cell virus, because
hmm,
even if the cells didn't have like the typical black look, I thought, oh, like non-viable queen cell, first thing I'm going to check is that pathogen. But we had pathogen screening done actually by David's lab,
yeah,
for the commercial producer who was having the problems too. Because when I had the problem, I was like just wanting to get queens to do my experiment, and like didn't want to mess around with figuring out exactly what was going on, because by the time I
yeah,
got data back, it'd be too late to get them from a local commercial producer anyway. So I just
yeah.
said, forget about it. I'm gonna go to this person. But then when they were having problems too, then I was like, well, maybe we should actually try to figure out what's happening.
So were
So
you getting cells, they just weren't viable, or was it they weren't
yeah,
making queens,
yeah,
period? Oh, they
cells that weren't
weren't
emerging.
viable.
Yeah,
What,
and
it
so
was black queen cell?
it was not, no,
Really?
that's I would have bet money on it being black queen cell virus. But we had the whole pathogen panel tested on them, and there was no difference between I think we had uh, nine or 10 viable queens, so like ones that emerged, and then
Hmm.
the same number of non-viable, and there wasn't any difference in pathogen level between the two. So
What? Are you
um,
serious?
yeah,
That's insane. Do you think it
so
was environmental then? I
yeah,
mean,
I
if I
think the
know if pollen
next,
flows, or if they if resources are shut off, it kills the mood, because I'll put like pollen sub out, and if they start wearing that out, I'm done. Like I just have to hang it
yeah,
up for a little bit, because
I
it totally dies off.
I believe this beekeeper was using homemade pollen patties.
Okay.
So like real pollen, not the sub.
Oh yeah, nice.
But I think the next sort of leading idea for what might have been happening to those queens is that it was either something nutritional, or perhaps some kind of pesticide
Yeah.
residues in the pollen, or somewhere in the environment that was affecting them.
Man,
Because
that's so bizarre.
there's quite a bit of research showing that that pesticide residues can impact queen viability, and by that I mean like in the developmental sense, not like the sperm viability, although there's that too.
Yeah.
um, so yeah, that was other work by Joe Malone from David's lab, that they reared queens in two different ways. They would rear queens in little wax cups that had
Hmm.
that were like impregnated with the
Coumaphos or something,
yeah,
or
exactly, with like
fluvalinate.
that blend of pesticides that's commonly found in wax,
Hmm.
and and compare those to ones reared in clean queen cups.
Yeah.
And then they also had tried feeding the builders pollen that was laced with the levels, like the realistic levels of these different chemicals found in the bee bread compared to ones that were fed clean pollen. And I believe it was that treatment that showed the biggest differences there in terms of the queen, impacting the queen development. But
wow, I'll occasionally find some, which again, it could be black queen cell virus. It seems like early season, and it's specific cell builders. Like I can nail it down to who it is once they start emerging, but like they're not black. You know how you have black goo a lot of times from black queen cell virus? It's not that. It's like they're stunted white pupae. Some of them will be worker size, like really bizarre looking stuff. Some of them just look weird.
yeah,
And I really would like to
was,
collect sample of those and just have a viral analysis run and see if it's something in particular.
yeah, well, it might show up with nothing, because that was similar
Yeah,
to
that
the
threw
ones
me off.
that I had here. Some of them were fully formed, just like stunted, like you
Dead.
say, and
Yeah.
no viruses.
Gosh, I wonder
So,
what, that is so bizarre. Do you think
yeah.
it could be? I guess it could be chemical. I'm in an agricultural area too, so it could be something they were exposed to. But again, it's odd because if I have multiple cell builders in the same yard, it'll be one in particular. That's why I'm assuming, heavy on the assumption that it's viral or bacterial, some sort of pathogen working on it. But
Yeah.
but
I don't know.
nutrition has a massive impact too, you know. So
Yeah,
there's
it's
so many
really hard to
variables.
say.
It is indeed. Alison, I know you're wanting to get to the barn and play with horses.
Yeah,
Do your
it's kind
thing.
of taken over my life now.
Yeah, that's all right. It's good to have hobbies. Bees are that for me.
Yeah, well,
Yeah,
here it's not exactly bee time yet.
it's true.
Yeah, we're sort of
True.
on the cusp, but I think later this week, there's one night where it's still going to get down to 0 degrees. So
Was it really?
yeah,
I
so
guess
we have
we're
these
seeing 19 tonight.
oh wow.
Well,
Wait,
19
Fahrenheit.
Fahrenheit.
What is
Yeah,
that? I don't even know.
it's a good question. It would be negative, right?
You'd think I'd know this by now, but it's
When
like it's one of those things that it's like I'm too old to learn new tricks or something. Like I can't,
I'm there. I'm totally there.
no matter
The
how
thing,
many times.
I just guesstimate. The only thing I know, and hopefully I'm not incorrect here, it's like 0 is freezing, right? And then what is 100? That's when water boils.
Exactly. Yeah.
Okay,
See,
that's
centigrade makes sense.
it does, but we're not gonna. We're Americans. We reject that.
Yeah,
It's ridiculous.
I try to have translatable temperatures. So like 4 degrees being the fridge, 20 degrees is like room temperature, body temperature is 37. So then with those,
Was it really?
yeah, with those, then you can kind of give people the idea of about like where other numbers fall.
This is somewhat related, but last time I was in Toronto, I got a rental car. I think it was like a Toyota Corolla or something, not fast or cool. And like I kind of got on it because I was merging onto the interstate. And I look up and I forget what my speedometer said, but I like freaked and hit the brake real hard. Then I realized it was kilometers.
Right.
I
Yeah.
thought I was like flying.
Nope.
Nope. I should have known. In the Corolla, it's not going to do that.
Yeah, that's right.
Well, I appreciate you taking time to catch up with me. I learned a lot.
Yeah, no problem.
I enjoyed it.
I hope that there's some useful information in here.
Oh, undoubtedly.
Yeah. Good stuff.
Thank you so much. Maybe we can catch up again whenever you got some more research coming out or something you want to talk about.
Yeah, sounds good. I don't have anything immediately in the pipeline, but I'm sure sometime this year some other stuff will come out. So
for sure. Well, thank you so much.
Yeah, thank you. Talk soon.
All right. Have a good evening.
Okay, bye.